Hi everybody, this video is going to be about aerobic respiration of glucose, and it's something that you're going to need to know for lecture and for lab. So you might remember this from high school, but just in case you don't and need a refresher, the steps in aerobic respiration are number one, glycolysis. You probably learned that the next step was the Krebs cycle, but that is not the way that Dr. Knight teaches it actually. So do you remember? So glycolysis is here, right? And then the Krebs cycle, and then there's like this little CO2 that squirts off right before the Krebs cycle starts. That is technically a separate step, which is called pyruvate oxidation. And so Dr. Knight teaches that as a separate step. And then last is the electron transport chain. So these are the 4 steps in aerobic respiration of glucose that you'll need to know, again, for lecture and for lab. Dr. Knight is going to have you learn the individual reactions in glycolysis, so you will also need that for your lecture test. We will not talk about the individual reactions of glycolysis in lab. So the main thing that is happening here is in these first 3 steps, you are dismantling the carbon thing that you have eaten in order to release energized electrons. You're going to capture those electrons in the electron carriers, the NADH and the FADH2, and then in the electron transport chain, you're going to make ATP, make a large amount of ATP using the energy that is currently being held in the NADH and the FADH2. So we're going to do a bit of an overview of this, and then we'll talk about each step in detail. So we're going to talk about this for glucose, but you guys learned in class that you also consume, well, you already knew, you also consume proteins and lipids. And so those will be broken down into their individual monomers, the amino acids and the fatty acids. And they do not enter glycolysis, but they will enter pyruvate oxidation and the Krebs cycle. And so even though we will learn this for glucose, it actually happens with every single food energy source that you eat. The other types of macromolecules will just enter at different points in the process. So FYI. So here we have a linearized version of glucose because I'm going to tear it into pieces, and I think it's easier to see if it is linear. Glucose can exist linearly or as a ring. And so normally when you see it in drawings in your textbook or whatnot, it's going to be in the ring shape. So in glycolysis, we start out with the 6-carbon glucose. And what happens in glycolysis is we break this bond in the middle. There's also some rearranging, but we break the bond in the middle to make 2 molecules of 3-carbon pyruvate. Every single time that a bond is broken, the electrons are released. So the bond is formed by the interaction of electrons. So when the bond is broken, the electrons are released, and the energy that is associated with that bond is now being associated with the electrons. And so it's really important for us to be able to make ATP that when we break the bond and the electrons with the energy are released, that they don't just go around the cell and stick to whatever. We need to carry them in a way that allows us to still use that energy. And so for that, we have 2 electron carrier molecules. There is the NAD+, which is going to carry 2 electrons and becomes the NADH. And then there is the FAD, which is going to carry also 2 electrons and becomes FADH2. So we're going to do a little side note on these, and then we'll come back to the carbon disassembly process. So the NAD starts with this plus right here. Can you guys see that? NAD plus. And so it carries 2 electrons. The first electron goes to counterbalance the initial plus. And so then the second electron allows an extra proton to bind. So NAD plus 2 electrons gives you NADH. FAD does not start with an existing charge. It's just FAD. And so when it carries the 2 electrons, each of those electrons can partner with a proton. And so it forms FADH2. So you've got NAD plus, which goes to NADH, and then FAD, which goes to FADH2. So just watch out that you don't get the number of H's on those mixed up. Okay, so in glycolysis, what we're doing is we're splitting the 6-carbon glucose into 1 glucose, 2 pyruvate, each of which are going to be 3 carbons. And so in the process of breaking this bond here, we are going to produce NADH because the electrons will come out, we will attach them to the NAD plus and make NADH. We'll go back through this and give you guys all the numbers, but this is just in general what happens. So now we have our 2 molecules of pyruvate. So, and that is this step right here. And so what's going to happen is you have your 3 pyruvates, and it is going to break into a 2-carbon acetyl molecule. And this is the same thing that is in acetic acid, vinegar. And then a coenzyme A group will come and attach to that. And so this is going to be acetyl-CoA, and this is what will enter the Krebs cycle. And then this 3rd carbon here, so acetyl-CoA has 2 carbons. This 3rd carbon here is going to be made into CO2. And so this is what we're going to breathe out. Once the carbon gets to CO2, we have extracted the maximum amount of energy possible. So we're just breathe that one out, throw it to the side. Because we have broken a bond here, we have also released electrons and captured those by an electron carrier, which is NADH. So then, in the Krebs cycle, I'll just draw it over here, we start out with our 2-carbon acetyl-CoA, and so we're going to completely break this down. So unsurprisingly, it is going to go to 2 molecules of carbon dioxide because we're going to completely break this here. The Krebs cycle is actually pretty complex. There is an existing 4-carbon molecule, and the acetyl-CoA sticks to it to make a 6-carbon thing, and then there's a whole bunch of rearranging. But at the end of the day, the acetyl-CoA breaks into 2 CO2s, and because there is so much rearranging, we actually are going to capture some electrons, as NADH, some electrons into FADH2, and then we are also going to make some ATP, which we also made up here, but I forgot to write down. So then in the electron transport chain, we are going to take every single one of our electron carrier molecules and use those to make, not directly to make ATP, we'll get back to this, but to power the formation of ATP with NADH and FADH2. So the first 3 steps are involved in completely dismantling the 6-carbon glucose into 6 individual CO2s, such that the bonds that were initially connecting the carbons are all going to be broken, and then those electrons will be released, and then we can use the energy from those bonds to be carried in the electron carriers and eventually use that energy to make ATP and electron transport. So again, first 3, complete oxidation, aka complete dismantling of the glucose into 6 individual CO2s. And that's going to be happen in the first 3 steps. And then the 4th step is making ATP using the energy and electrons from the NADH and the FADH2. So I'm going to stop this video and then make you guys another video that has more details.